Today we're going to be talking about essential concepts of brain function in dementia. Anybody who cares for a victim of dementia has to understand the basic principles of how the brain works. The objectives for this session include, number one, identify important brain regions and their functions. Number two, distinguish the function of neurons and synapses. Number three, describe brain development. Number four, explain neurotransmitters and neuromodulators. Number five, identify brain locations that control memory, language, motor skills, sensory recognition, social skills, moods, and temper. Number six, understand the differences between the voluntary and the involuntary motor systems. And seven, explain differences between the brain, mind, and soul. To begin with, what is it that distinguishes our brains from those of other species? Well, the human brain contains 13 billion neurons. That's billion with a B. And if you look at this next graphic, above you see a normal human brain, and below you see the brain of a monkey. What distinguishes the human from the monkey is not the shape or the gyri, but rather the number of neurons. It is the large memory capacity, the large computing capacity of the human brain provided by those 13 billion neurons that allows us to do complex functions such as high-order social skills, abstract reasoning, and language skills. Brains come in all sizes and shapes, and the size of the brain doesn't necessarily indicate greater or less intellect. For instance, in this graphic, on the left you see an extraordinarily large brain. The average human brain weighs about 1,350 or 1,400 grams. The brain on your left weighs 1,750 grams. It was from a normal individual, not a genius. The one on the right weighs 1,300 grams, which is a normal size. Sometimes people think that the kinds of changes that occur in the brains of individuals as they grow older are remarkable. If you look at the brain of a normal elder, as seen in the next graph, you see that there aren't that great a change. The brain on top is from a 45-year-old individual. The brain down below is from a 75-year-old individual. It is slightly reduced in volume. However, the overall size is not drastically reduced. Likewise, if you look at this on coronal section, coronal meaning being cut right through the center, the top is the 45-year-old individual. The bottom is the brain from the 75-year-old individual. It is true that as you grow older, your brain becomes a little bit smaller. However, you do retain most intellectual functions as you grow older. Let's begin by talking about the surface anatomy, the neuroanatomy of the human brain. To begin with, we want to look at the surface anatomy and then consider the microscopic anatomy, and finally consider the chemical anatomy. Each of the different anatomies is involved with a different kind of disease, and you will be taking care of many of these patients whose level of damage may be at the surface, such as in a stroke, at the microscopic level, such as with Alzheimer's disease, or at the chemical level, such as delirium or depression. The brain is separated from the skull by a thick fibrous covering called the dura. The next picture shows this white dura lying on top of the brain after the skull is removed. And the picture after that shows the dura folded away, exposing the gyral surfaces. A large vessel called the sagittal sinus runs down the middle and collects blood from the brain through bridging veins labeled in the next slide as BV into the sagittal sinus as labeled by SS. Sometimes blood vessels that run between the dura and the brain are severed, and this leads to a subdural hematoma, as seen in the next picture and labeled by SDH. This large blood clot sits between the brain and the dura. This mass of blood will compress the brain, as seen in the next slide, with the normal brain on the left and the compressed brain on the right. Subdural hematomas are fairly common in patients with Alzheimer's disease. The next picture shows the lateral surfaces of the brain, which is divided into three general areas: the cerebral hemispheres, the brainstem, and the cerebellum. Each fold or gyrus on the outer surfaces of the brain has a name. For instance, the arrow points to the middle frontal gyrus. The brain section on these coronal slabs, as seen in the next slide, demonstrates the middle frontal gyrus here that is bounded by the superior and inferior frontal sulci as located here and here. The cortical ribbon, or gray matter, as depicted by the arrows, is the tan-brown tissue on the outer surfaces that overlay the white matter, which is deep inside the brain and is composed of axons and dendrites. The gray matter is composed of neurons. These gyri almost touch in normal brain tissue, and the sulci are quite narrow. In the section below, from an Alzheimer patient, the gyri have shrunken, the sulci have widened, and this is referred to as atrophy, a fairly common finding. Ventricles are fluid-filled cavities deep inside the brain. The lateral ventricles stretch beneath the frontal, parietal, occipital, and temporal lobes, while the third ventricle is situated deep inside the brain. The fourth ventricle is located inside the brainstem. Cerebrospinal fluid is produced in the choroid plexus, located within the ventricles. This clear liquid flows through the third ventricle, out through the fourth ventricle, up and around the brain, and is finally reabsorbed in structures embedded in the blood vessels of the dura called the arachnoidal granulations. The loss of brain tissue in Alzheimer's diseases causes these ventricles to become dilated, as seen in the next picture, where you see normal ventricles up above, as depicted by the arrow, and down below the dilated ventricles seen in Alzheimer's disease. As we see in the next graphic, the human brain is divided into lobes. There is the frontal lobe, the parietal lobe, the temporal lobe, where a lot of memory and language is stored, the occipital lobe, which is involved with vision, and then beneath the cortex is the brainstem in the cerebellum. We're not going to talk very much today about the cerebellum. It is involved in balance and other fine motor activities. However, in most dementias, the cerebellum is not affected. However, the brainstem is, and we'll spend a lot of time considering that. Let's move on to the microscopic anatomy of the brain. And in the next slide, you see what a nerve cell would look like under the microscope. The arrow points to a neuron in the 11 o'clock position. In fact, there are multiple neurons on this field. The processes going off from each end are the axons and the dendrites. The blue material in the midst of the cell body is the protein machinery or the Nissl substance. This is a higher magnification of a neuron. You see that the body has a triangular shape, and there are all of these processes stretching out like limbs of a tree. These are the axons and the dendrites. Information flows in through the dendrites and out through the axon. At a higher magnification, you can see there are all of these little outpouchings from the dendrites. These are the specialized areas of connection where the axons from other nerve cells touch and connect with this individual neuron that you see on your screen. In the next slide, we see nerve cells that actually contain a brown pigment called neuromelanin. These nerve cells produce special chemical transmitters such as norepinephrine and dopamine. We're going to talk about those in a little bit. Neurons come in different sizes, different shapes, and have different materials stored within them. When you're talking about the neuron at the cellular level, you have to understand the difference between the axon, the dendrite, and the synapse. These key features of the neuron are described in the next several pictures. Again, the neuron has three major parts: the dendrite, the cell body, and the axon. The information flows in from the dendrite and out via the axon. Every neuron has one axon that branches off to contact thousands of other neurons in the brain over specialized regions called synapses. The next drawing shows what a synapse on a dendrite would look like under higher magnification. Thousands of axons from neighboring neurons form these specialized contacts with the dendrite called a synapse. Each of these four swollen areas with roughened surfaces represents a type of synapse where information is transferred from one cell to another via chemical neurotransmitters. If we look at these areas under even higher magnification, we can see that there are many different types of synapses. The message is sent from the axon to the dendrite via neurotransmitters, or packets of chemicals released from the axon into the synaptic space, where it flows across the space and lands on a receptor which is designed to recognize only that specific brain chemical. Eventually, a second chemical removes the transmitter from the receptor and recycles the molecule. This drawing shows the three major types of synapses: excitatory, inhibitory, and modulatory. A fourth type of messenger, called the trophic factor, also has a specific receptor. The electrical impulse travels down the axon and causes the presynaptic membrane to release packets of chemical neurotransmitters that float across the synaptic cleft and attach to the specific receptor on the postsynaptic membrane, where the receptor produces some type of chemical or electrical change in the dendrite. Excitatory transmitters, here, cause the dendrite to fire. Inhibitory transmitters, here, lessen the chance of firing. And neuromodulatory transmitters control the rate and likelihood that the dendrite will fire. Some synapses are linked to channels, which are pores in the lipid membrane that allow charged particles to flow in and out of the cell. Each dendrite has thousands of messages arriving simultaneously, and the nerve cell has the ability to condense these signals into a single message. Most psychoactive medications alter the release of neurotransmitters, or receptor systems that receive the chemical messages. Once the chemical message lands in the receptor, the neuron releases substances that remove the message from the receptor and recycles the chemical. This is the 3 R's of neurotransmission: release, reception, and recycle. Some chemicals, like antidepressants, increase the amount of transmitters that is released, while others block the recycling so that the chemical sits on the receptor and stimulates the neuron for a longer period of time. Others alter receptor activity. You have to understand your 3 R's in order to understand neurochemistry. Which then brings us to the molecular level. You have transmitters, these chemical messengers that go from the axon to the dendrite. You have receptors, the specialized area where the transmitter is received. And then you have a third category of molecular messengers called trophic factors, which we're going to talk about in just a little bit. Once you get into the cell itself, at the subcellular level, within the individual nerve cell, there is what is called the cytoskeleton. This is a delicate meshwork of filaments and tubules that can't even be seen with the microscope. You have to use an electron microscope. The cytoskeleton is responsible for the structure of the nerve cell, its structural integrity, as well as transport, the movement of vital substances within the individual neurons. Cytoskeleton is important because some scientists believe that it is an abnormality of the production of this cytoskeleton that may lead to Alzheimer's disease. In addition, there is the membrane, this outer lipid skin of the nerve cell, in which most of the electrical activity is transmitted. And then there are channels. Channels are specialized pores that exist within the skin, the membrane. And these channels allow the movement of charged particles such as sodium and chloride and potassium. And it is the movement of these charged particles that causes the electrical activity within the individual nerve cells. Channels are important because chemical transmitters may affect the flow of ions through these channels, and some medications affect these channels and their control of the ionic flow. If you look at neurochemical substances that are important in the brain, the 3 important ones to know are excitatory, inhibitory, and neuromodulatory. There are 3 types of neurotransmitters that you absolutely need to understand in order to know how the brain works: excitatory, inhibitory, and neuromodulatory. Excitatory transmitters are those which cause the nerve cell to fire. Inhibitory transmitters are those that lessen the likelihood that the nerve cell would fire. And neuromodulatory transmitters don't cause the nerve cell to fire or not fire, but rather control the rate that it will fire at. What are some typical examples of excitatory transmitters? Well, amino acids. These chemical substances may be important in disorders such as stroke and ischemia. Inhibitory transmitters include the substance gamma-aminobutyric acid, also referred to as GABA. This is a compound that is affected by drugs like Valium and Librium. Neuromodulatory transmitters include chemical compounds such as serotonin or norepinephrine. Trophic factors include things like nerve growth factor, the substance that's being studied in Alzheimer's disease. If we're going to talk about neuromodulatory transmitters, the majority of these compounds are produced in the brainstem. The brainstem is that part of the brain which sits underneath the cortex. And in this next picture, you see a depiction of what the brain would look like if it were cut down the midline. You see the brain region here underneath the corpus callosum, referred to as hypo, standing for hypothalamus. But beneath that is a part of the brain called the midbrain, as depicted by mid. Pons is the second level of the brainstem, and medulla, or MED, is the third level of the brainstem. Midbrain, pons, and medulla are the three major regions of the brainstem. The brainstem controls the centers that manage things like your blood pressure, your pulse, and your respiration. They also control the centers that produce important catecholamines. In the next slide, you see the location of important catecholaminergic centers. Catecholamines are norepinephrine, dopamine, and serotonin. The SN stands for the substantia nigra. That's where dopamine is produced. The LC stands for locus ceruleus. That is where norepinephrine is produced. And the R stands for raphe. That's where serotonin is produced. Dopamine, norepinephrine, and serotonin are three of the major neuromodulatory transmitters that control a myriad of functions in the brain. And those are the centers that are damaged in aging, Alzheimer's disease, and Parkinsonism. In the next slide, you see levels to your right through the midbrain and to your left through the pons. The brownish area, as depicted here by the arrows on the right-hand side through the midbrain, is where the substantia nigra is located. These nerve cells produce dopamine. When they die in Parkinsonism, you become stiff and begin to tremble. On the left, the arrow points to nerve cells in the locus ceruleus. These neurons produce norepinephrine, a chemical transmitter which is essential to both memory and to mood. These nerve cells die in Alzheimer's disease as well as in aging, which is why depression is common in individuals over the age of 65 and why patients with Alzheimer's disease have a high rate of depression because of noradrenergic deficits. Serotonin is made by nerve cells that sit in the midline. These pigmented nerve cells can be seen under the microscope as brown dots, as seen here. Each one of these produces norepinephrine. These cells are from the locus ceruleus. So here you can see, again, that same brainstem level with the production of dopamine as depicted by DA, norepinephrine as the NE, and serotonin, the 5-HT, the levels at which each of these important chemical transmitters are produced in the brain. The brainstem basically is where your brain's thermostat is located. These neurons control the firing rates of other nerve cells up in the cortex, as seen in that convoluted area up above. What are some important neuromodulators? Well, acetylcholine, which is made by nerve cells in the base of the brain, is important for cognition. When it becomes deficient, you develop confusion. Norepinephrine is important with depression, as is serotonin. Dopamine is important for movement and is the chemical that is deficient in Parkinsonism. Histamine is important for sedation, which is why antihistamines cause people to become drowsy. And GABA is important for anxiety. We've talked about lobes of the brain, we've talked about neurons, we've talked about synapses, and we've talked about transmitters. What is the pathology that is associated with damage at each level? Well, for instance, when you damage an entire lobe, as what you see in a stroke, then you lose an entire function. For instance, the ability to move one side of your body. As opposed to if you just have damage to neurons, as when you see in Alzheimer's disease. There you get patchy focal loss of function. As opposed to if there is some disorder at the synaptic or transmitter level. If that happens, then you have what is called delirium, which is temporary global dysfunction. Or if there's a disruption of the transmitter system, such as norepinephrine and serotonin, that leads to depression, which is a temporary, patchy, or global dysfunction of the brain. Each of the different levels of anatomy has a different associated pathology and a different associated dysfunction. Instead of looking then at discrete brain regions such as frontal lobe or neurons or brainstem, let's talk instead about brain systems. If we're going to talk about brain systems, and there are many, the ones that we need to focus on in order to understand dementia and the management of patients with dementia is the motor system, the sensory system, our information storage systems, in what often people refer to as the limbic system or the emotional system. To begin with, let's talk a little bit about the surface anatomy of the brain. Here you see the lateral view of the brain. Notice the infoldings, the cauliflower appearance to the brain. That is to increase the surface area because it is on the surface where most of the neurons are located. Now, these surface areas are divided into lobes, and different lobes have different functions. So on the next cartoon, you can see the breakdown of different functions. We call this compartmentalization of the brain. Different brain regions do different things. So it's possible for one area of the brain to be damaged and you to lose one function, but the other area of the brain to remain intact, and that function will be retained. So, for instance, in the frontal lobes over there, you see that mood and speech, social graces are programmed, as opposed to the motor strip that we're going to look at a little bit more carefully, where voluntary motion is programmed. Understanding and hearing is programmed in the temporal lobe, as opposed to recognition and eyesight, which is in the occipital lobe, to the back of the brain. And arousal and attention are driven by nerve cells in the brainstem, as seen in the structure underneath. Let's focus on the motor system as an example. It turns out that you have two different types of motor systems. There is the pyramidal and the extrapyramidal. You have one motor system that tells your hand to move based on your thoughts. You have a second motor system that controls muscle tone and posture without you ever thinking about it. The pyramidal system moves your extremities on mental command. The extrapyramidal system controls posture and tone without you ever thinking about it. That's broken down then into a voluntary and an involuntary motor system. The pyramidal or voluntary system is located in the cortical motor strip. The extrapyramidal or involuntary motor system is located in deep brain nuclei. For example, if we look at this lateral view of the brain, the pyramidal system is located on the outer surfaces in what is called the motor strip. It is very highly organized so that control of your mouth and your lips is to the bottom of the motor strip, as opposed to your trunk and your legs, which is on the top of the motor strip. The extrapyramidal system is located deep inside the brain. Here we see a view through the middle portion of the brain, cut in the same plane as you would see with your hat. The arrow points to the basal ganglia and the caudate nucleus. These deep brain centers are the seat of the extrapyramidal system that controls tone and posture, as opposed to the pyramidal system, as seen here by the new arrows, that points to the cortical or voluntary motor system. When these deep brain structures are damaged, patients become Parkinsonian or develop tremors, as opposed to the superficial system, where when those are damaged, the patient loses the ability to voluntarily move their arms or their legs. The next slide depicts the relationship between the brainstem, as seen here by the arrow, the substantia nigra, as seen by this second arrow, and then the pyramidal or cortical system, as seen here by the arrows on the surfaces. The brainstem nerve cells located in the substantia nigra in the midbrain are part of the extrapyramidal system. It is damage to these nerve cells, these neurons, that lead to Parkinsonism. And of course that makes sense. Patients with Parkinsonism develop stiff, shuffling gaits, can't move their faces rapidly as they want to. They oftentimes develop a tremor. On the other hand, they can still voluntarily move their arms and their legs. That's because the voluntary system is spared, but the involuntary system is damaged. In order to understand why Parkinsonian patients can move but can't maintain posture or tone, you have to understand the difference between voluntary versus involuntary. Therefore, when you start talking about damage to these systems, in the pyramidal system, strokes are a common pathology. What that leads to is paralysis, but oftentimes the patients remain stiff. That's because the extrapyramidal system still works to a certain extent, as opposed to the extrapyramidal system, in which Parkinsonism is the most common pathology. And in these individuals, although they can still move their arms and the legs, they develop abnormal movements, shaking and stiffness. Pyramidal versus extrapyramidal, those are the motor systems. Let's talk a little bit now about sensory systems. There are three parts to your sensory system. There is the special sense that pulls in the information. That would be, for instance, your eyes and your ears and your nose. There are the subcortical pathways that relay it to the cortical interpretation centers. So all sensory information has to stop in a deep brain region called the thalamus. That's important because some patients with injury to their thalamus may tell you that they feel pain, even though there is no pain. It's called thalamic pain. So sensory information comes in through the special senses, goes to the thalamus, and then it arrives at the cortex where it is interpreted. The one we're going to focus on today is hearing. Let's think a little bit about the organization of the sensory cortex associated with hearing. There is the primary sensory cortex, where the sound of my voice is imprinted in your brain as a series of electrical clicks. Then there is the unimodal association cortex, where those series of electrical clicks are translated into words that are then compared to words that are present in your stored dictionary in order for you to arrive at some meaning as to what I am saying. And then there is the heteromodal association cortex, which is a higher order association cortex where not only the words and their meaning is being interpreted, but also the context in which they're being said. For instance, if you're walking down the street and somebody says, "Are you having a nice evening?" that has one meaning. On the other hand, if you're walking down a dark street in the middle of the night and somebody sneaks up behind you, some rather large individual, and says, "Are you having a nice evening?" that has a totally different meaning, doesn't it? That different meaning is being discerned in your heteromodal association cortex. The interpretation of the words, in other words, what are the words actually meaning, is being deciphered in your unimodal association cortex, and you are hearing those words in the primary sensory cortex. In fact, you have one part of your brain that speaks and another part of your brain that hears. And it is possible to be able to talk without being able to hear or to understand. That is fairly common in stroke patients in those individuals with Alzheimer's disease. Here in this graphic, on the top you see a normal brain, and in the bottom you see the expressive area, what's called Broca's area, depicted by the talking. That's where the motor activity of speech is generated, as opposed to the Wernicke's area, where your receptive cortex is located and the temporal lobe is situated, where you hear and understand. It's possible to speak but not understand, and it's possible to be able to understand but not be able to speak. They are two separate functions compartmentalized in different parts of the brain. If you have a patient who seems to be able to talk, don't assume that they can understand, and vice versa. Because here in this next picture, you see what happens in temporal lobes of patients who are normal versus those of individuals with Alzheimer's disease. The first arrow here on the left points to what's called the transverse temporal gyrus. This is where you are hearing what I say. The second arrow here points to the superior temporal gyrus. This is where you understand what I'm saying. This is the unimodal association cortex. The third arrow here points to the temporal pole. This is where everything is being interpreted together to give the words some meaning. What you see on the other side in the Alzheimer brain, as the arrow points here to the transverse temporal gyrus, is that the cortex looks pretty much normal, similar to that of the normal on the left, as opposed to here and here for the superior temporal gyrus and the temporal pole, which is the association cortex, which is severely atrophic. This patient can hear but not understand, because the hearing part of his brain is left intact. The understanding and the synthesis part of his brain has been damaged by the disease. Different parts of the sensory cortex do different things, and therefore different areas can be damaged, leading to a broad range of deficits. Let's move on and talk about information storage. How is it that you can remember things? Well, there are 5 basic steps for information storage. First of all, you have to be able to identify incoming data. Secondly, you have to be able to get that data loaded up. Thirdly, when you've got too much data loaded on the system, you have to discard it. Fourthly, you have to decide what you're going to store. And then once it's put into storage, you have to be able to retrieve it. These are all functions that are served by different brain regions. Let's talk a little bit about memory. You have 2 different kinds of memory. You have recent and remote memory. Recent memory is things like what happened 5 minutes ago, where you left your car parked. Remote memory is those things that happened 5 or 10 years ago. Recent and remote memory. Recent memory is a discrete circuit located in the temporal lobe. It is affected not only by the neurons in that circuit, but by neuromodulators. Remote memory is located over extensive neural networks in the temporal, parietal, perhaps occipital cortices, and is also affected by neuromodulators. Recent memory is programmed in the mesial temporal cortex in structures such as the hippocampus, the amygdala, and parts of the temporal lobe. Remote memory is stored over the entire cerebral cortex. Therefore, it's harder to erase remote memories than it is to disrupt recent memory. This next slide depicts a temporal lobe from a normal individual up above and down below the temporal lobe of a patient with Alzheimer's disease. The arrow points to the hippocampus above. This is the gateway to recent memory. Nerve cells in this brain region decide whether or not information is going to be put into long-term storage. In order to get information into long-term storage, you not only have to have these hippocampal nerve cells functioning properly, but you also have to have a proper balance of neuromodulators, such as norepinephrine and serotonin, affecting the performance of those nerve cells in the hippocampus. So it's possible to disrupt recent memory by two different mechanisms. One is to kill the nerve cells in the hippocampus so that information can't flow through. And the second is to disrupt the neuromodulators so that those intact nerve cells don't work right. Down below, you see an example of an Alzheimer's hippocampus. Notice how shrunken and atrophic it is. That's why patients with Alzheimer's disease have significant short-term memory impairment. They can never remember where their car was parked or what you told them 5 minutes ago because this part of their brain is dying. Likewise, in depression, although the hippocampus is intact, the norepinephrine and serotonin, which is important for those nerve cells to function properly, is deficient. That's why patients with depression sometimes have memory problems as a part of the depression. Not because these hippocampal neurons are dying, but rather because the essential neuromodulators are out of balance. When you fix their depression, their memory deficit improves. Unfortunately, there's no way to fix the damaged neurons in Alzheimer's disease, and their recent memory impairment is fixed. The best way to understand how your brain processes information is to consider how your computer processes information. In fact, your brain is like a huge PC and has all the components of that machine that sits on your desk. The typical PC has 4 major parts: the hard disk, the workstation, mechanisms for loading and unloading information, and built-in programs that tell the system how to run. Your brain has each component located in discrete regions. You upload data into your system through special senses like hearing and eyesight. You download data through the abilities to communicate with speech or writing. You have a workstation that allows you to consider specific pieces of information, and this is located at least partially in the temporal lobe, while your hard disk stores long-term memory, and this is located over the cerebral hemispheres. Your built-in programs are located in the brainstem, which turns the system off and on and tells the computer how fast to run. Consider as an example how your brain tries to remember all the information that is presented on this tape. Your eyes and your ears gather the data and present it to your brain. Your sensory cortex assembles the information into a meaningful message which is located onto your workstation where you think about this material. When your brain decides what message will be stored, the information passes through the hippocampus, which is the gateway to your hard disk. The information is loaded onto your hard disk in the cerebral hemispheres and assigned a location so that it can be retrieved. Your brainstem or your system's program control how fast the information can be loaded onto or off of the system. Your brain, like your computer, can malfunction. It receives millions of pieces of information every day. It does not have the capacity to store all those facts and experiences. Your mind has the ability to distinguish between important or trivial information and stores significant facts. Your brain has a save or file button just like your computer. Patients with Alzheimer's disease have malfunction of their save button. Even though their find button still works and they can still retrieve old files, patients begin to lose the ability to store new information. Returning to the previous slide, we see a temporal lobe from a normal individual and that of an Alzheimer patient down below. Notice the shrinkage of the hippocampus. This patient can load and recognize information in the system, but they cannot store it on the hard disk, even though they can retrieve old memories from the hard disk. When a person's workstation malfunctions, they lose the ability to concentrate, or they get wrong information, such as with patients with mental illness who perceive false sensory input or have false ideas. These are referred to as hallucinations or delusions. Depression is caused by brainstem malfunction that slows the system down. The brain as a PC, it's a pretty good model to understand how these systems work. What are some factors that impair the human memory? Well, fatigue is one. We've all experienced that. The more tired you are, the less able you are to remember. Medications, anything that disrupts this circuit in the hippocampus, whether it be cause dysfunction of the nerve cells located there or the nerve cells that produce neuromodulators, such as acetylcholine and norepinephrine, can cause memory problems. Illness can impair memory, severe anxiety. Sometimes the circuit's running so fast you can't get anything through it. And brain damage. Many types of brain damage result in injury to those circuits that program memory. Moving on from information management, let's talk a little bit about the limbic system. People oftentimes hear about the limbic system and the limbic lobe, and it comes from the Greek word limbus, meaning boundary. The limbic system or the limbic lobe is actually not a discrete part of the brain, but it is actually a collection of connected areas that bounds the brainstem. It surrounds the brainstem, which is why it gets its name limbic or limbus, or bounding the brainstem. It's sometimes referred to as the Pepez circuit. It's important to remember that the limbic lobe is not a lobe like the temporal lobe or the frontal lobe, but rather it is a collection of interconnected brain regions that also receive extensive catecholaminergic innervation that is involved with emotions, memory, and cognition, as seen in this picture with the brainstem disconnected from the brain. When we remove the brainstem, as seen in the next picture, the labels point to major components of the limbic system. L1 is the amygdala, L2 is the hippocampus, L3 is the cingulate gyrus, and L4 includes the fornix and the mammillary bodies. These interconnected structures form the limbic lobe. Emotions are controlled by other brain regions as well, especially the inferior frontal cortices, as seen here by the arrow. Limbic lobe function then involves emotions, memory, and certain levels of cognitive functioning. But again, it's important to remember that this is not a discrete anatomical region, but rather a collection of interconnected brain areas. Now that we've talked about not only the surface anatomy and the microscopic anatomy, but also the functional organization of the brain, let's talk a little bit about developmental stages. First of all, it's important to understand development because you will come to recognize the fact that most dementias are actually development being played in reverse. How is it that our brain develops in such a fantastic way that we are able to store all of these memories and perform all of these intellectual tasks? Well, there are 5 basic stages in development. First of all is what's called neuralization. That is where the nerve cells, those 13 billion neurons, reproduce to the point where there is a sufficient number to populate the brain. The brain cells pretty much stop reproducing within the first 3 months in utero. That's right, within the first 3 months of uterine development in the fetus, the brain cells have pretty much completely reproduced. Once you are born, nerve cells will not reproduce. What you have is what you get. That is why whenever a nerve cell is damaged and the information is erased, it's difficult to reprogram other nerve cells to do that because they do not regenerate. All neurons begin in the middle of the brain and have to wander to the outer surfaces or the gyrus or the cortical ribbon, and that is called migration. Once they get to the cortical ribbon, they must then integrate themselves with all of the surrounding neurons. That's referred to as synaptic integration. And then those neurons that are not wired into the network properly are supposed to be programmed to automatically die off. Once the neurons are wired into the network, and those nerve cells that are incorrectly connected have died back, then in the final stages in adolescence, there are numerous modulations of neurotransmitters, so that by the time our brain reaches the age of 19 or 20, it has gone through an amazing sequence of developmental events. Above, in this picture, you see a normal human brain. Below, you see the brain from an individual who would have been about 30 weeks gestation. Notice the difference in the size. The other thing that you notice is that the surfaces don't look as convoluted, as folded in the infant's brain or the neonate's brain as in that of the adult brain. And in fact, if you look here, the brain in the bottom is a 20-week, the brain in the middle is a 30-week, and the brain in the top is a 40-week. So you go 40, 30, 20. What you also notice is that the more immature the brain is, the less folded it is. It is this folding or gyration that allows for increased surface area. You need a broad expanse of surface area in the human brain because that is where the neurons are stored. And having 13 billion neurons on the outer cortical mantle means that you need a lot of surface area. So these gyri produce increased surface area that allows the neurons to be present where they need to be. Here you see the distinction between a 20-week below and a 30-week above. This is development. Neurons traveling from the middle of the brain to the outer brain and then arranging themselves in the network where they need to be. In fact, on the left you see a normal hemisphere, and on the right you see a hemisphere from a 4-month-old infant. And in fact, it looks like just a slightly smaller copy of the adult brain. Basically, at term, at 40 weeks when you're born, your brain is pretty well developed. This is an amazing thing. Think of what this involves. It would be as though every telephone in the United States were placed in a warehouse in Kansas City, and somebody needed to write a computer program that made those telephones migrate on their own from that warehouse in Kansas City all over the country and land directly in your home on your nightstand. And then once that telephone got on your nightstand, it automatically connected itself into the network and was a right number. And if it wasn't a right number, then it automatically disconnected itself and took itself off the network. What an amazing sequence of events, neural development. Sometimes the brain does not develop normally, and when that happens, we have what are called developmental disorders. 3 common examples of developmental disorders include mental retardation, such as Down syndrome, schizophrenia, and autism. Let's talk a little bit about mental retardation. The brain on the left is that of a 4-year-old individual who was profoundly mentally retarded. Notice how small it is in comparison to the brain on the right, which is that of a normal individual. This patient was mentally retarded because of an insult to their brain in utero. And as a result of that, many of the neurons were killed before they arrived and were wired into the network the way they were supposed to be. There are a broad range of injuries to the brain that lead to mental retardation. Of course, the distinction between mental retardation and dementia is dementia is the loss of intellectual functions that you previously had. Mental retardation is that you never had the intellectual functions to begin with. This is a second brain from a profoundly mentally retarded 2-year-old who suffered severe birth asphyxia, resulting in near total loss of all neurons. The brainstem, however, remained intact, so their respiratory centers survived and they were able to sustain their life. On the other hand, there are sometimes subtle changes in the brain. On the left is a normal brain. On the right is that of a patient with Down syndrome, trisomy 21. This individual was able to dress themselves and tend to their own needs, but had a functional IQ between 50 and 60. Notice that the brain is somewhat smaller than that of the normal brain on the left. This is typical of what you see in many mentally retarded citizens, whose brains oftentimes look normal. So then the question gets to be, well, what's wrong? Well, what happens is that the nerve cells get to where they're supposed to be going, but they don't get into the correct position. Or if they're in the correct position, they don't get connected into the network they're supposed to be in. Or if they get connected in, they are a wrong number. Imagine what it would be like in our telephone system, for instance, if every other time you dialed the number, it was a wrong number. We'd have a hard time communicating, wouldn't we? And if there are lots of wrong numbers in neurons in the brain, then there is a lot of dysfunction in the communication from one neuron to another. Or imagine what it would be like if all the neurons didn't get into their correct location and simply malfunctioned. These are the kinds of things that can happen in the brains of mentally retarded people. The same thing happens in dementia, but only in reverse. Imagine what would be like to try to communicate in a telephone system where every year millions of telephones became disconnected. That's what's happening in dementia. Millions of neurons are dying year in, year out. This is a different kind of developmental disorder. The temporal lobes below are from a normal individual. The temporal lobes above are from a patient with schizophrenia. We now know that patients with schizophrenia have abnormal brains. Remember I was saying that the temporal lobe is the part of your brain that does your hearing and your understanding. Well, we now know that the temporal lobes of patients with schizophrenia are not normal. We think maybe they are abnormally wired, which may be why patients with schizophrenia have auditory hallucinations. They hear voices when the voices are not there. It has to do with the fact that they've got neurons that are not connected properly in their temporal lobes. The same thing occurs, by the way, in degenerative diseases like Alzheimer's disease. About 25% of them develop hallucinations. And again, this is the system malfunctioning. And in fact, when you look at the brains of patients with Alzheimer's disease, here's what you see. This is a mid-sagittal view of three brains. The top is that of an individual who is normal. The middle is that of a patient with Alzheimer's disease. And the bottom is that of a brain from a patient who is about 16 months old. What you notice is that the middle brain, the Alzheimer brain, looks more like the infant's brain than it does like the adult's brain with regards to size. And that has to do with the loss of millions and millions of neurons as a result of the disease process. And we talk about Alzheimer's disease and other types of dementia as being development in reverse. As you learn things, they are added in layers. For instance, speaking. You learn to speak in single words or phrases, and then you speak in simple sentences, and then you learn to speak in paragraphs. What you're doing is your computer is being programmed through learning, and you are developing increasingly sophisticated language skills. What happens in the dementias is that those language skills that you learn through development begin to reverse themselves as the neurons that store that information are stripped out. So first you lose complicated language skills, then you lose simple language skills, and then eventually you lose all language skills. We talk about Alzheimer's disease and other dementias as being development in reverse, because if you were to make a live video of all of your intellectual functions and play it in reverse, it would be very similar to the clinical symptoms that you see in a patient with Alzheimer's disease. Development in reverse. Let's move on to the highest order of intellectual function, what I like to refer to as systems integration. Things like judgment, behavior, emotions, and wisdom. These are very high-order intellectual functions which require the use of all of your cortical and subcortical intellectual abilities. These functions are easily disrupted by brain injury, whether it be through trauma, stroke, or through Alzheimer's disease. Things like behavior, judgment, and emotions involve a complex mixture, as we see in the next slide, of cortical, as seen by the cortex above, and subcortical, as we see by brainstem below, functions. These functions are harder to localize because of the sophisticated intellectual nature. However, on the next slide, we see the listing of some different areas that are assigned to each of them. For instance, emotions. We know limbic structures as well as catecholaminergic innervation are involved in control of our emotions. Behavior, impulse control, for instance, sexual behavior, involves limbic structures, deep brain or diencephalic structures, and catecholaminergic innervation. Judgment involves particularly frontal lobe function. Patients with significant frontal lobe injury oftentimes make serious mistakes with regards to their judgment, act in an impulsive way, and get themselves into trouble. Wisdom is an even higher order cortical function involving the entire brain function. No one has yet to identify the brain region specifically assigned for wisdom. Now let's talk a little bit about how the brain repairs itself. We spoke earlier about the fact that nerve cells will not reproduce. So when a nerve cell dies, it's gone, and the information which is stored on it is erased. Part of this is protected against because your brain is sort of like the space shuttle. It's got redundant systems. So if one circuit that stores memory is erased, then there are multiple other systems that are still in place that serve that valuable function. Eventually, though, in diseases like Alzheimer's disease or stroke, enough of the systems are damaged that you lose the function. Sometimes the neural network will reorganize itself, and we see that in individuals who, for instance, may have a big stroke in one part of their brain and may shift some function to the other hemisphere or to another part of the brain. The third thing is what is referred to as plasticity, the ability of the brain to rewire itself. The brain does retain, even into the 7th and 8th decade, a limited ability to rewire itself. And it does that by its ability to extend the dendritic arborization. Those are all of those synaptic and dendritic processes that stretch out and touch other neurons. Those have the ability to increase in number. We also have the ability to increase the number of synapses, the connections between individual neurons. And thirdly, we have the ability within our brain to increase receptor efficiency. In conclusion, the aging brain retains some ability to repair damage from disease. The human brain maintains its capacity to recapture function even into the 8th and 9th decade. These three mechanisms allow the brain to use alternate circuits to perform tasks previously controlled by damaged neurons. Researchers and pharmaceutical companies are studying ways to promote regeneration and reinnovation within the human brain. Older individuals may help promote these mechanisms by maintaining an active intellectual life. Finally, let's talk a little bit about spirituality. This is an area that we who are neuroscientists know very little about. It's important to understand, though, that there is a distinction between the brain, mind, and soul. Because so many times people misunderstand symptoms of brain and mind malfunction for a disorder of the soul. Let me give you an example. If you go to the museum and see a beautiful painting, your brain, your visual cortex, looks at that beautiful painting and compares it to all of the images that you've seen before and comes up and says, oh, that's the Mona Lisa. It's your mind that interprets what that Mona Lisa means. Where is the history for this? What is its significance? But it is your soul that is moved by the beauty and the symbolism of this great art. Brain, mind, soul. In most of the diseases that you're going to learn about in subsequent segments on dementia, we're dealing with people whose brains and mind are severely damaged by disruption and death of neurons, loss of catecholamines, loss of receptors. But it's important to remember that all of those things have nothing to do with the soul. And even though their brain and their mind may be severely damaged, their soul is still in there.